Now, after secondary folding, some proteins are going to get folded more, and they're going to take on more of a globular or three-dimensional shape. Enzymes and other bigger proteins tend to move to this tertiary structure. In the tertiary structure, it's created based off of the interactions of the R groups. So secondary structure is the interaction of the backbone to get it kind of organized, and then tertiary is the R groups based off where they're pointing. They're going to interact. That is why we needed to classify and identify the polarity of our R groups so we can then predict the interactions that are going to occur. If we have two nonpolar groups interacting, what's the strongest force that can happen in a nonpolar interaction? I already have it written down for you. So dispersion forces, right? We're only going to have dispersion forces interacting between nonpolar R groups. Dipole-dipole forces, we're going to need a polar group like a sulfur or our amides that can then interact with our dipole interactions. We can have hydrogen bonding with any of our alcohol groups. We can form disulfide bonds. This is pretty unique to proteins. What a disulfide bond is when we have those thiols. So thiols are an SH group. We don't really worry about them at our level because this is the only time we see them. So in cysteine, they both have a carbon group and then an SH. Those SHs react together, very similar to like an ether, and they're going to create disulfide bridges or bonds, so two sulfur atoms. It's a physical covalent bond that is formed, and so it's really the most rigid one that we can have. And then those ionic interactions between the acidic side group and a basic side group, creating those ions, having that electrostatic force. What essentially occurs is once we have our secondary folding, right? So we have some folding here, and then we've got some just unfolded chain of polypeptide, and then we have our beta pleated sheet, and then some more on parts of the amino acid or the protein that doesn't have secondary folding, and it just starts to come together into a blob. You like my blob? So it comes together as a blob. The only way it can do that is to have those R group interactions so that it can stay. My pipe cleaner does not have R group interactions so that it can't stay together. On a more mature note, visually what we can see is based off of those R groups, what can we identify, right? So these are the different interactions that we can form. Here we've got that amine group of one amino R group and that carboxylic acid. So we have that positive and negative charge forming those ionic interactions. Below, or the next one over, we've got that hydroxyl group with that carboxylic acid. And so a hydroxyl group with a carboxylic acid is going to form that hydrogen bond. Next, with these phenyl groups, you can see that the phenyl groups are interacting here, but because they're both phenyl groups, that's going to be a nonpolar. And so with that nonpolar, that's going to give us our dispersion forces. Same with this guy here, all those interact. We have the disulfide bridge. And then we can have interactions between the backbone and R groups and water, but it's the purpose behind seeing how these R groups interact, why we need to be able to classify them, so we can understand how it's going to fold and orient itself, both within the protein, but also with its surrounding environment, which you'll see in your proteins to cell membranes activity. Now I know my pipe cleaners did an amazing job showing you this three-dimensional direction. No, it didn't. That's okay. This is how we typically will do a computer-generated protein. What is really neat about this is you can see how we have these arrows. So these yellow arrows, these are the beta pleated sheets. These purple are going to be your alpha helices. And these, what would have been my brown pieces of my pipe cleaner, those are just unfolded segments of the protein. They don't have specific secondary folding, but they can interact with that tertiary folding based off that. And typically we will find arrows to help us understand in this ribbon drawing of that sequence order as well.